Okay, I think we're going to get started here. Just reminder once more to mute your mic and turn off your camera if you can. It helps keep the stream looking nice for everyone involved. So I am Diana Boxma. I am a steering committee member on the Grassland Restoration Forum. I've been a member for a few years now, and it's just a lovely group. So part of what this lovely group does is we present webinars to people like yourselves. So thanks everyone for taking the time to join us. Today's webinar is the first in our winter webinar series, and it's on the topic of herbicide usage in invasive non-native plant management. I'm just going to give you a little bit of update on the GRF and a few things that's coming up, and then we'll get into our talk today. So the Grassland Restoration Forum's mission is to provide a forum for information sharing, tool development, research, and education. And we do that to promote and support conservation and effective restoration of native grassland ecosystems in Alberta. So to help support those goals, we offer this free webinar series. So today we have Victoria Wagner presenting, and then in February, Emily Holden with ABMI will be presenting. Her topic is the function of plant-plant interactions in community dynamics of native grassland. You can register for that one now on our website. In March, we're going to have Sean Asselin of Agriculture and Agri-Food Canada, and he's going to give us an update on what he presented last year, and that is in regards to his research in breeding native plants adapted to our area. And then in April, we're going to have John Acorn, who is the nature nut, as you may know. He's with the University of Alberta, and he's going to speak on conservation in eroding landscapes. So do pay attention to our website and our email outs, and registration for the March and April ones will be up very soon. The February one you can register for today. So one of our annual courses that we offer is our Range Health Assessment Training course. We offer that in the fall in Brooks as a 2-day course, with day one being an in-classroom look at how to use the range plant community guides and how to use the recovery strategies manuals. And then day 2 is more field-based. We go out and actually do that work. What we found is September is still field season, so day 1 often has to get canceled due to low registration. So we are going to offer that course March 26th at the Glenborough Ranch House. And it's just that standalone day 1 of how to use the range plant community guides and those recovery strategies manuals. It's really useful training. So if you're going to be doing any grassland work this year, native prairie DSAs or pre-disturbance site assessments, putting together seed mixes, any type of assessments in native grasslands, super valuable course. And the really nice part of this is it's a good chance, because it's in March, it's just pre-field season. So it gets your brain working about, these are what the plants look like. These are what my communities are going to look like. It's a really nice course, not just for new people, but also for more experienced people. So do consider taking that one in. You can head over to our website right now and register for it. So it's March 26th at the Glenborough Ranch Schoolhouse in Cochrane. And then you can also always look for our September course will be announced soon. Okay, so do again, we've got lots of webinars coming up. We offer courses. This presentation is going to be recorded, so you can go online and look at that one. We also have our webinars from last year are recorded and online. So yeah, just head to the link where you registered for this one and you will find all the information that you need. So with that, let's get into our topic today. So just a few rules of engagement. So as I mentioned before, please mute your mic and turn your camera off. If you have any questions, you can add them to the chat bar. We're going to save those till the end, but feel free to post your questions at any time. And then Ross Adams is also a steering committee member with the Grassland Restoration Forum, and he's our behind the scenes guy. He's been letting you in and making sure this presentation runs nice. So thank you, Ross. He is going to manage the chat and read out the questions at the end. So he'll make sure he's got it. Yeah, and then if you're not, if we weren't clear on the question you were asked, or if there's more to add, we'll give you the chance, raise your hand and we'll let you unmute at the end and we can either clarify or you can ask a new question then. So I'm going to introduce Victoria now so she can get going. So Victoria Wagner is a plant ecologist and associate professor in the Department of Biological Sciences at the University of Alberta. Her research lab uses field surveys and experiments to understand patterns and drivers of non-native plant invasions, mechanisms of seed dispersal in prairie grasslands, and plant diversity of rare habitat types. She has published 3 book chapters and 50 research articles, including contributions in invasion ecology and invasive species management. Victoria is going to speak to us today about studies on herbicide use in greenhouse and grasslands of Montana and what we can learn about efficacy and non-target impacts. Go ahead, Victoria.
Thanks so much for the introduction and invitation to talk about my research, specifically my research on herbicide usage in invasive non-native plant management. And I'm going to share some of the results of our research on patterns of herbicide use, herbicide efficacy, and non-target effects. And yeah, I'm an associate professor in the Department of Biological Sciences at the University of Alberta. And I'm showing here the logo of the University of Montana at the bottom of the slide, because this is when I initiated my research on herbicide effects as a postdoctoral fellow at the University of Montana in Carl Nelson's Restoration Ecology Lab. And the research that I'm going to talk about used the foothills grasslands of Missoula and surrounding areas as a study system. I just wanted to acknowledge that this land is traditional land of the Salish, Xanka, and the Clispe peoples. And in fact, the name Missoula is derived from the Salish name Mesuletque, which is the place of freezing water, and likely refers to Glacial Lake Missoula. The research was conducted by a whole set of collaborators. I wanted to mention Kara Nelson, who was my host at the University of Montana, and we've been collaborating on herbicide effects now for over 10 years, not as actively anymore as we used to do, but we're still in touch occasionally. And then Christine McManaman and Rachel Lugar were 2 undergraduate students who were very productive, Christine at the University of Montana and Rachel at the University of Alberta, who either conducted whole experiments or helped us with complex data analysis. So thanks so much to Christine and Rachel. And I'm Pedro Antunes, so professor at Algoma University in Ontario, and Michael Irvine worked for the Ontario Ministry of Natural Resources and Forestry, and they were involved in our herbicide usage estimate study.
Sorry
Victoria, can I just
Victoria.
get you? Yeah, can I just get you to turn your camera off? And I think we're just seeing a little bit of blurriness on the visual.
Oh, my camera?
Yeah, I think
I have to unshare. Okay, hold on.
I might be able to turn your camera off, actually.
Turn my camera off. Here we go. And then I'll. Okay. Can you see my slide?
Yes.
Okay, perfect. Just want to get my laser pointer back. All right. Yeah. As mentioned in introduction, my lab focuses on non-native plant invasions. We have quantified levels of invasion across different habitat types in Alberta. We studied the invasiveness of certain plant groups, specifically agronomic invasive grasses, and recently started focusing on Cargana arborescens as an escaping and invading shrub in the southern boreal forests of Alberta and Saskatchewan. And another recent project is seed dispersal ecology in the dry mix grasslands of Alberta. And yeah, we do field surveys, experiments in the greenhouse in the field, and we work with existing data sets. Because of the detrimental impacts that non-native invasive plants can have on native plant communities and ecosystem functioning, many countries and regional administrations have put in place legal frameworks to control and regulate invasive plants. So in Alberta, we have the Weed Control Act, and in the United States, many land managing agencies also have documents that mandate invasive plant management. And practitioners have a different set of tools at their hands of how to manage invasive plants. These comprise mechanical tools like mowing and pulling, biological methods, fire, and chemical controls, which are herbicides. And although all these tools are used, there's little information on how frequent or common these tools are being used. A recent literature review by a group from Brazil found that chemical means comprise 42% of interventions that was studied in scientific literature. So if we assume that the scientific literature, the applied scientific literature, reflects what's going on in ecological practice, then we can say that herbicides are likely the most common tool that practitioners use to control invasive non-native plants. Now, they also looked at methods per continent and looked at the frequency of chemical versus non-chemical tools, which you can see in this bar plot figure here. And they found that in general, the more wealthy regions were, the higher their usage of chemical interventions was compared to non-chemical interventions. So because herbicides are so commonly used, it's important to study their efficacy and non-target effects. And I wanted to briefly introduce the different herbicide types that can be used. I know probably many of you in the audience know the differences, but I just wanted to sum up the different herbicide types just for those in the audience who are not familiar with herbicides. We generally differentiate between two groups of herbicides. Selective herbicides are those that target specific groups such as broadleaf plants. Tordon and Milestone are two commercially available formulations that include active ingredients, which in this case are picloram and aminopyrrolide, but they are in their commercial formulation, they're mixed with all kinds of other additives such as surfactants to help herbicides penetrate plant tissue more efficiently. And then we have a group of non-selective herbicides, which target plants no matter what group they belong to. And Roundup, which contains glyphosate as an active ingredient, is an example of a non-selective herbicide. Roundup targets an enzyme pathway that is that's happening in all plants, which is why glyphosate is a non-selective herbicide active ingredient. And in my talk, I'm going to present 4 papers. First, I'm going to talk about an estimate of how much herbicides are used in invasive non-native plant management. Then I'm going to talk about the effects of herbicides on plant germination. And third, a paper that explored the role of spray timing and seeding. And the fourth one will be a more complex paper that resulted from a field experiment where we looked at the long-term efficacy and non-target effects of 2 selective herbicides on foothill grassland plant communities. When I started my postdoctoral fellowship at the University of Montana, having finished my PhD, I was very excited to get started on a more applied topic. And I wanted to look at non-target effects and efficacy of herbicides in invasive plant management, because I felt there's so much research on invasive plants, but there's very little, compared to that, very little research on invasive plant management. So I teamed up with Kara Nelson, who was also very enthusiastic about the topic. And one of the first things we wanted to address is how much herbicides are actually being used for invasive non-native plant control. Now, when I started, there was good data available for weed control in croplands. So that's released at the level of country and also globally, but nothing comparable was available for non-native plant management outside of croplands and outside of recreational areas such as golf courses and residential areas such as your backyard. So I embarked on a study to it was the first estimate of herbicide usage in that particular context. And I was a little bit naive. I have to confess that I didn't expect it would be that much work, but I pulled it through, and I contacted various agencies and organizations in Canada, the United States, and Mexico, and also got in touch with agro-statistical companies because they often compile data on herbicide usage, at least in crop lands. So in addition to my, I asked whether data is being tracked and whether the agencies or organizations would be willing to share that data, and whether they have any data on monitoring of herbicide effects. And although I contacted so many offices, it was a lot of work by email and phone. Only 5 responded that they actually track the data, the overall land managing agencies in the United States, and only 4 shared the data with me. They included the Fish and Wildlife Service, the National Park Service, the Bureau of Indian Affairs, and the Bureau of Land Management. I'm very thankful for these agencies for sharing that data with me. So what I did is once I received the data, I cleaned it and I standardized it and combined it to provide an estimate of herbicide usage. Now, the data that I received from these agencies span different years, and I used the year 2010 for an estimate because this is the year for which I had the most complete data from all agencies. And what I found was that in terms of the amount of active ingredients used for invasive plant control in natural ecosystems, a total of 201 tons of active ingredients were used in 2010. And just to give you an idea of how much that is equivalent to about 27 London double-decker buses. I also looked at land sprayed and calculated that 0.5 million hectares of natural land were sprayed, which is about equivalent to about 462,000 soccer fields. Now, when I calculated these numbers, I found they were huge, but then I compared them to what is being used in cropland. So when I looked at the amount of active ingredients used, I realized that what I calculated for natural lands was just 0.1% of what is being used in cropland. So yeah, big numbers for invasive non-native plant control in wildlands, but it's just a tiny amount of what is being used in croplands. We were also interested in the monitoring data, and most agencies that shared the data with us had some entries on monitoring, but the data were restricted most often to 1 or 2 years, and the data was also not standardized, so it was very difficult to analyze it. So we didn't look into that, but I think it's an opportunity if agencies could have a standardized protocol for monitoring and entering the data. There could be an opportunity for the agencies and also researchers to look at effects and efficacy. When we broke down herbicide usage by active ingredient, glyphosate was by far the most common active ingredient used, both in terms of air treated, which is shown here on the left side, and amount used, shown on the right side. And we believe the reason for this high usage by glyphosate, we believe there are several reasons that could explain that. The patent for glyphosate expired in 2000, which made it cheaper to purchase. Glyphosate does not require a license to spray in most states. Glyphosate is also available in some formulations for aquatic use. So these are some of the reasons we think why glyphosate is so popular in invasive non-native plant control. But it is a non-selective herbicide, so it can potentially harm desired native plants. Now, we also conducted several experiments to look at non-target effects of herbicides because we were concerned about the effects of herbicides on desired native plants. And in the Missoula area, a common practice is to spray a herbicide and immediately follow up with seeding of desired species. So we wanted to see how this practice affects germination of native plants. And we focused on two selective herbicides, picloram and aminopyralid, which are commercially known or sold as Tordon and Milestone, because these are the most common herbicides. And they're specifically are used in the Missoula area to manage knapweed, Dalmatian toadflax, leafy spurge, among other invasive forbs. At the start of the experiment, we prepared our soil by sterilizing a sand-low mixture in order to kill the seeds that were present in the soil. And then we applied herbicides with a backpack sprayer outside of the greenhouse using a recommended rate as stated in the manual for the product, and a diluted rate. And the diluted rate was used in order to simulate drift for herbicides to nearby non-sprayed vegetation. And then there was also a water control. And after we sprayed, we returned our pots back into the greenhouse and added seeds of 14 species, 50 seeds per pot, and we had a total with 20 pots per species. We had, I think, 5 pots that we lost, but in total we had 275 pots in our experiment. For those of you interested in the types of species we used 5 native monocots and 2 native dicots, which are common in the foothills grasslands around Missoula. And we wanted to compare the response of native species to non-native species. Just for the sake of comparison, we didn't expect there to be any biological differences because herbicides are there is no herbicide specifically target non-native plants. And we found, we looked at the response of each species. I'm not going to go through all of the graphs for every species. I just picked up some that represent overall trends that we saw among species. So in general, we found that picloram, both the recommended and diluted rate, and aminopyralid and the recommended rate suppressed germination. And you can see that in this graph here. This shows the response of Cerevignaria spicata and Idaho fescue. It shows the number of emerged seedlings per pot on the y-axis and the different treatments on the x-axis. The control is on the left. The 2 aminopyralid treatments here are in the middle here, and picloram is on the right. So yeah, you can see here that picloram really suppressed all the germination, and no matter what rate it was used, and the same thing applies to aminopyralid in the recommended rate. The diluted rate of aminopyralid did not have a strong effect compared to these other treatments. The effects varied by species, but we did find that the only 2 native forbs that we included, Erigeron speciosus and Geranium viscosissimum, were very sensitive to herbicide application. Their germination was completely suppressed for herbicides, no matter what rate was used. And we did not find any difference between native and non-native species. So here are results for Timothy and leafy spurge, which showed that they're also sensitive, just like the native species, to picloram and aminopyralid in the mentioned concentrations. Now, the experiment I just presented was really very artificial, you might say. So it was in a greenhouse and using pots. And we wanted to understand to what extent these detrimental effects of herbicides that we observed on seeding can be translated to more realistic setting in the field and more to a framework that's or practice that is used by practitioners. So this is where Christine McManaman's study comes in. She did this amazing study consisting of actually 2 experiments. in which she looked at the timing of seeding after spraying and how it affects germination and early growth. Because we predicted that the longer the time between the spraying and seeding, the less likely it is that herbicides will have a detrimental effect, because herbicides leach through the environment and they're also broken down in the soil. Christine used 10 species for her greenhouse experiment listed here. All of them are common in the foothills grasslands of Missoula. And the ones with an asterisk are those that she also used in her common garden experiment. So she worked only with native species. And she used a similar protocol that I used for her first experiment in the greenhouse. But she included only 3 treatments, picloram and aminopyrrolate, again, our 2 selective herbicides, but only in the recommended rate and water as a control. And she had 5 timings between spraying and seeding. The very first batch of pots without any seeds was sprayed in the fall, and these pots had a time of 11 months before seeds were added. And then she had a timing treatment of 9 months between spraying and seed addition, 6 months, 3 months, and 0 months. And the latter refers to an immediate seed addition after spraying. She used 360 pots for every timing batch, which resulted in 1,800 pots. I just wanted to say that I've supervised many students, and this number is really amazing, especially for an undergraduate student. So she did an amazing job with her project. She also followed up with a common garden experiment where she sprayed in the fall, again using aminopyrrolate, picloram, and water control, and sprayed in spring, again using the same treatments. And then Christine added seeds in April. So we have basically 2 timing treatments between fall and mid-spring, and a very short timing for the spring. This was a common garden experiment that she carried out using 72 plots. And after seeds were added, Christine counted the emerging seedlings and assessed the seedling biomass. She produced this long paper, and I just wanted to highlight some of her findings. The greenhouse study results are shown on this slide here. The very upper graph is a bar plot that shows mean relative difference in percentage. And you can see that the numbers here are all negative, and they refer to pots sprayed by herbicides and their number of germinants or biomass as compared to the control. So because these values are negative, you can already see that herbicides had a negative effect compared to the water control. And then the x-axis lists the time in months between the herbicide application and seed addition. And interestingly, we don't see any trend in timing. So timing apparently did not matter in her greenhouse study, no matter how many months the pots, no matter the timing between the spraying and seed addition, herbicides did suppress native plant germination, which was quite interesting, and it could be explained by by different causes. For instance, that in the greenhouse, the breakdown of the herbicide and the leaching of herbicides might have not taken place as compared to field conditions. And again, similar to my experiment, Christine did pick up different signal among species, which is shown here in the table. I will not go into details, but we do see across our experiments that some species seem to be more sensitive to herbicide application at the seed stage than others. for her common garden experiment, that was an interesting outcome, and it was different compared to the greenhouse experiment, a little bit different. She found that 75% of species showed adverse effects in the spring, whereas the fall spring was not as detrimental. Only 25% of species were affected by that. You can see this here in the figure, which shows the number of germinants on the y-axis for two species that I'm showing you here, Gaillardia aristata and Pseudorygnera spicata. And then the right column shows biomass, seedling biomass. And then the treatment is shown here on the x-axis with fall treatments showing on the left and the spring treatment showing on the right. And then the different herbicide treatments are displayed as different symbols, aminopyrrolate, picloram, and control. So in the fall, you can see there's not much difference between herbicides and control, probably because there was more time for herbicides to be broken down or to leach into the environment. It's really that short-term spray and seeding that had more detrimental effects, as shown here. And again, picloram had a more severe effect than aminopyrrolate in some species. So we can summarize that Christine's study showed that she confirmed that when you study effects in the greenhouse, there's a very strong negative effect of herbicides on seeds. But then when you look into field condition, the response is less pronounced. We do see negative effects, but timing matters. And the field response seems to be more complex, also based on other factors, species differences. And we also believe that there might be differences due to soil types and litter present in the sites.
Sorry.
So we then conducted another study to investigate effects under more realistic conditions using a field study. This is our most realistic study I can talk about. And I initiated the experiment as a postdoc with CARE, and then collected the data over several years. And Rachel, who was then an undergraduate student, helped me to analyze this complex data set. And we addressed two study questions. First, we wanted to know what is the efficacy of herbicides, or how effective are they in reducing the target invasive plant, and are there possible non-target effects on desired native plant species and the community. We hypothesized that because the herbicides we were studying were forb selective, that graminoids would be the winners in our study, and that we would see secondary invasions by invasive grasses in our plots. And there was a parallel study at the time conducted in the Missoula area by Ylva Leckberg, and I'm not going to talk about the study because there's not much time, but what she found was that when knapweed was targeted with herbicides, what happened was that once knapweed was suppressed, invasive grasses moved in, annual grasses like bulbous bluegrass, and the other one was cheatgrass. So we hypothesized that something similar might happen in our plots as well. And then we hypothesized that forbs would be the losers in this experiment because we're using forb selective herbicides, and that both native and non-native forbs would be equally negatively affected. The experiment was carried out at 3 different sites in and near Missoula, and again in foothills grasslands, with 30 plots per site, which resulted in 90 plots in total. And our target species was spotted knapweed, very common in the Missoula area. And we chose sites that had intermediate levels of knapweed invasion. We specifically avoided knapweed-dominated sites because we wanted to have some native plant species and plant communities to also study their response. This is how our design looked like. We set up a big area of 24 by 20 meters and used a so-called fully randomized design where we had smaller quadrats of 2 by 2 meters that we randomly place in this matrix. And we installed wooden stakes that lasted over several years and used 5 treatments, the same treatments that I also used in my greenhouse experiment. They comprise Tordon and Milestone and the recommended rates, as well as their diluted rates to simulate drift and a water control. There were 6 replicates per treatment in site. I initiated the experiment in the spring of 2012. This is when I did a pre-treatment survey of all plant communities. This is the 0 point in the study. I then sprayed the plots in August of 2012. And we did 3 post-treatment surveys, one in 2013, one in 2015, and the last one was conducted in spring of 2018. And we were specifically interested in change in Centaurea cover, and we estimated foliar cover of all the other species present in the plot in order to assess changes in species richness and species sum cover for the groups we were interested in. I'm first going to talk about the results, the outcomes for the efficacy in suppressing the target plant knapweed that's shown here on the left. This figure here shows the cover of knapweed, and the x-axis show the year of our study from 0, which is my pre-treatment survey, and the last year, which was the 6th year of the study, shown here on the very right. And the different treatments are shown here in colors and symbols. The water is shown in gray. Tordon treatments are shown in orange, and the milestone treatments are shown in blue. So we expected that our herbicides would actually work. And what we saw, however, in the first year was that there was an overall drop in knapweed cover, including in the water control. So which was unexpected, and we believe it has to do with an unusually dry winter in the months before we conducted the field work. So, and we think that this dry spell overrode any herbicide effects that we expected. But then in the 3rd year, we did pick up a signal, and we saw that the 2 recommended treatments with Tordon and Milestone did significantly suppress knapweed compared to the water control and the 2 diluted controls. So that, yeah, this is where the expected treatment happened. But then, interestingly, by year 6, all these differences, the herbicide effect was gone, which emphasizes the importance of long-term monitoring. If we would have conducted the study a year after or 3 years after, we would have gotten very contrasting impressions and a different would have made different inference. Now let's look at the effects on species richness, again using similar graphs here, now with species richness shown here on the y-axis. And what we saw interestingly was that in the first year we did pick up a signal with respect to our the entire total the total species richness per plot, with Tordon having reduced species richness in the treated plots significantly compared to especially the water control and the diluted treatment. So Tordon seems to be detrimental to species richness. We did not find any difference in graminoid responses with respect to species richness, respective whether they were native or non-native. And then native forbs did show a signal, specifically native forbs, and they seem to be quite vulnerable to herbicide application when in the case of the recommended treatment. So both Tordon and Milestone led to a decrease of native forb richness in the first year, but then in the 6th year, differences among our treatments disappeared. So there seemed to have been like a splash, an effect in the first year, but then differences disappeared in the longer term. And although some trends emerged from non-native forbs, they were not significant. With respect to species cover, we did not see any differences among our treatments. Cover did drop in the first year. Again, we believe this is due to the dry spell in that year, in the previous months, and then cover recovered over the years. We thought that graminoids would be a winner and that there would be in particular secondary invasions by invasive annual grasses, but this was not the case. So graminoids, graminoid cover did not differ between herbicide treated plots and the water control. And similarly, non-native graminoids did not display any secondary invasions. And this might have to do with the fact that our plots were not dominated by knapweed. They had a mixture of native plants, and that the native plant richness that was present at these sites was able to buffer invasions by annual invasive grasses. Then similarly, forbs were not a loser. Forbs did not respond to our herbicide treatments, except for non-native forbs, which showed a weak response to herbicides with a decline due to the recommended rates of tort and milestone in the 3rd year. Now, this result is quite interesting, and we think it might also be the result of species responding differently to herbicide usage. When we looked at some common plant species, so we tried to identify plants that were present in almost in all the plots, so where we can see how they responded. And when we look at the species level, we do see that some species respond sensitively to herbicides, such as Lupinus sericius, which showed in the 3rd year a negative response to the Tordon recommended application. And similarly, Aculeum was sensitive to Tordon and milestone, the recommended rates of these herbicides, and its cover was reduced in the 3rd year too. And these 2 species were able to recover by the 6th year. So unfortunately, because we used established plant communities, we can't really provide more insights into species-specific effects. I think you really need to have a common garden experiment where you specifically plant a species that you're interested in and then assess the response, and hope that such studies could be carried out in the future. So to sum up the research that I've conducted with my colleagues, we can say that herbicides are commonly used in invasive plant management, but the magnitude of usage and their effects are rarely tracked. Herbicides can impair native plants at the seed stage, but the effect is generally weaker and more complex in the field setting. We also saw in the last experiment that I talked about that short-term assessments of herbicide effects can be misleading, and what we need is really long-term monitoring of non-target effects and also efficacy. And that, like from a perspective of the researcher, I think the most interesting field to look at or aspect to look at are species-specific responses, because it appears that some species are more sensitive than others. And we also believe that the soil type affects how communities respond to herbicide usage. Yeah, with that, I would like to finish my presentation and thank the Humboldt Foundation for funding my postdoctoral fellowship, as well as my an NSERC grant for helping me finish my herbicide research field sampling in Montana. and all the technicians and collaborators who enabled this research. Thanks so much for listening.
Thanks, Victoria. I'm going to let Ross, if you're ready, you want to get into the questions?
Yes, yeah, I'm just gonna scroll back here. Apologies, folks. The network issues were certainly not foreseen, so thank you for bearing with us. But feel free to throw any other questions in the chat. And okay, so first one is with most invasives being plants that humans and livestock can consume, what is the reliability of simply eating our invasive plants into extinction? Maybe that wasn't a serious question, but if you do want to answer that, you can take it away there, Victoria.
No, I think that's a serious question that has been debated among researchers to what extent it's ethical to consume invasive plants. I think it's more question of ethics, morale. Yeah, I think different people have different opinions. I'm more on the side of we can consume and we should consume, seeing no issues there.
All right, the next one, is there data on germination success if native plant seeding occurs a couple months before herbicide application?
Yeah, so that's what Christine McManaman's study looked at. So she looked at how herbicides applied in the fall affect germination when seeds were added a few months later in the spring. And yeah, she showed that if you space it out in time, the herbicide application versus seed addition, then the effect seems to be less detrimental.
Okay, next one. Do you plan on continuing this study to examine the effects of herbicide on nematodes, mycorrhiza, and soil-bound insects?
Oh, I think these are great aspects. I'd love to. As a plant ecologist, I feel this would be beyond my expertise, but I can say that I collaborated with Ilva Leckberg on the effects on mycorrhizae. I didn't talk about this paper because I thought I would not have much time, but she looked at mycorrhizal responses, and I looked at the vegetation response. So we combined our efforts here. And as far as I remember, she found that I think I would have to look at the paper. It's been a while ago, but I think she found that knapweed is a good host for mycorrhizae. And when knapweed is eliminated through herbicide application, the mycorrhizal abundance in the soil decreases too. So we shouldn't assume just simply that, that invasive plants are also detrimental to soil biota. In this case, they are, let's say, a beneficial host to mycorrhizae, and that when they're eliminated, there might be a decline in soil biota abundance, a beneficial soil biota abundance.
Alrighty, next one. Were drought conditions and plant stress levels considered when looking at the vegetation response to herbicide application?
We did not include that as a variable, like we did not formally study that aspect because we did not see the outcome coming. But in retrospect, we can only speculate that a dry spell in the very first year after herbicide spraying in our field experiment is probably responsible for the patterns that we saw. And the patterns were, no herbicide effect whatsoever, no efficacy, all the different metrics dropped, richness and cover. So yeah, I think to address that, you really want to set up a field study that spans many more years, and then so that you can get that range of different precipitation patterns. Yeah, but it's a very important aspect, I think, when thinking about herbicide application is how was precipitation the previous months and also after application. Yeah.
All right, next one. What do you think the relative effect of seed bank composition or vegetative infilling from edges to promote infill of native species?
Yeah, I think it's very important. We didn't study that aspect, but I think it would be great if a study could look at herbicide effects on the seed bank and also on the depth of seed location and soils to see whether maybe seeds are drilled into soil, or maybe the existing seed bank can buffer negative herbicide effects during application. I think that would be very interesting. I don't remember the second aspect that the question referred to. Oh, infilling. Yeah, absolutely. I think the matrix of the entire vegetation is important to take into consideration. In our field experiment, as I mentioned, the the knapweed invasion was not very severe. So I think that could also explain why we see our plant communities that were sprayed bouncing back to the initial state. I think in settings where the invasion is more severe and larger, that response could have been very different, and we might have seen more secondary invasions by other non-native plants. Yeah.
Alrighty, a couple more here. Are there any new herbicides or herbicides currently in development that show promise to decrease the impact on native plant communities?
I have to say, I don't know, because I'm currently not actively pursuing herbicide research. That would be a question, I think, for somebody else in the audience who is maybe more up to date with new herbicide trends. I'm sorry.
Oh, fair enough. Are there any impacts on the propagule success of plants in the community impacted by herbicide usage?
Could you repeat that question?
Are there any impacts on the propagule success of plants in the community impacted by herbicide usage?
So I understand this question refers to the field study. We did not add any seeds, so I can't say what the fate of seeds was in that experiment because we only assessed the above ground standing vegetation. But I think a field study that uses seed addition and herbicide spraying would be very interesting, and that would be really the ultimate realistic study. Yeah, we haven't conducted that, but in general, seed addition experiments are in the field are complex and they're also risky because you have so much going on in the field with seed predation. Yeah, I think that would be a very different undertaking.
All right, last one we've currently got. How successful are herbicides on non-native graminoids such as smooth brome or crested wheatgrass?
That's a really good question. I think based on the literature, so it's not something I've studied in my research, but based on literature, I can see that there is a variable response with respect to herbicides. I know that there's been some studies reporting that other applications or interventions such as spring grazing might be more beneficial. And I know that there are also groups that study specifically the usage of fire to control agronomic invasive grasses in prairie grasslands. Yeah, but I can't tell you like specific trends that I recall seeing in the literature.
Alrighty, that I think gets us to the end of the questions, and we're coming up to time here. So, thanks, Victoria. Did you have any concluding remarks there, Diana?
I just have, reminder to head over to our website and sign up for the next presentations that are coming up in February and March and April. And also on our website, you can sign up for that March 26th Range Plant Communities Training Course in Cochrane. And, also while you're on our website, we have lots of great resources. So do peruse over there. So, and just a reminder that this will be posted on our website, and we'll be sure to post the slides with it because I know they weren't clear during this whole presentation. So we'll make it work. Okay, I think with that, we can let you all go. Thanks again, Victoria.
Thanks so much.